of the entire symposium. So our second keynote talk and closing presentation will be given by Dr. Aashish Manglik of the University of California, San Francisco. Prior to opening his own lab at UCSF, Dr. Manglik received both a PhD and MD from Stanford, where he worked with Dr. Brian Kabilka and published several impressive articles on GPCR structures, including of the beta-2 adrenergic and mu opioid receptors. Some of his more recent research as an independent investigator has looked at understudied and unconventional receptors, employing structural tools to better understand signaling behavior, and it's some of this work which I believe he will be showing us today. Dr. Manglik is widely recognized as a rising star in the GPCR research field, and we're honored that he was able to join us as a keynote speaker. So let's all thank him and listen to what I'm sure is going to be an exciting close to this symposium.
Great. Thanks so much, Ian, for the introduction, and thank you all for inviting me to this really exciting symposium. It's really great to see a lot of names of trainees and people that I know, and hopefully we'll get a chance to connect with a few of the others. So what I want to do today is tell you a little bit about work that's ongoing in my lab. Let me just share screen here. Hopefully that looks correct. Trying to understand a variety of very unconventional GPCR stimuli. And hopefully what you've gotten a sense for over the past couple of days, as part of the symposium, is that at this point, the ability to use structural biology and biophysical methods to interrogate GPCR signaling has come incredibly far away, certainly since when I started in the field about 15 years ago. But it turns out that many very unconventional stimuli for GPCR still require kind of a confluence of not just structural biology, but other methods. And that's what I'd like to give you a flavor for today. So just to set the stage, I know this is all of you are here literally for a GPCR conference, but I think it's maybe worthwhile to underscore why we all remain so excited and why this field is such a large field. It's because the single family of proteins in the genome, so again, the single largest family of proteins in the genome, is responsible for sensing such a huge diversity of stimuli. And this is what for me keeps me perpetually fascinated by this field. How can this one family of proteins recognize everything from hormones, proteins, ions, pH, light, or force? And as you heard beautifully from Sumit just in the last talk, such a huge diversity of stimuli. How does this work, and how does that all then impinge on a common architecture for these proteins that then leads to intracellular signaling cascades? Now, there's a huge diversity of GPCRs. This is just a dendrogram, a nice dendrogram made by Ray Stevens and colleagues now about a decade or so ago, showing about 400 or so non-olfactory GPCRs in the genome. And as many of you know, the really fascinating thing about these receptors is they control almost every aspect of physiology. And so regardless of what area of biology you're interested in, it's likely there's a GPCR that participates. And that's what perpetually brings new people into our field, which is what's so exciting. Now, a big goal of the structural biology era for the past 10 years has been to understand what allows information transfer to happen faithfully across many GPCRs, and how does that break down in disease? And I like to really think about GPCRs as effectively cellular channels, right? They're channels on which information is broadcast. And what's amazing about the structural biology era is that we now understand at a deep level a chemical logic that enables faithful signal transmission across many of these channels. And so this is a very kind of perhaps overused analogy, but for a long time, predating the structural biology era, that the notion was to identify, if the receptors are locks and the keys are their ligands, which keys go to which locks. And of course, for many receptors, so-called orphan receptors, we still don't know what the keys to those locks are. But for the structural biologists and the structural biology, what's provided us is a lens into how the keys actually impinge on the locks to open them. And on the one hand, this is great for doing stuff like drug discovery. Obviously, it has great implications for that. But the other side of this is that if we can understand this kinds of principles, I think we could also perhaps use that to perhaps open up areas of GPCR science where we actually don't understand what the key is. And some of the stuff that Sumit presented probably starts to get at notions around that. So I'll give you two examples of unconventional stimuli that my lab has been particularly interested in lately, and our use of not just structural biology, but new methods to try to interrogate how these receptors work. And the first one that I want to tell you about today is a really unconventional stimulus, which are protons. So it turns out that among the 400 or so non-olfactory GPCRs, 3 receptors in particular are really tuned both at a kind of a very in vitro level, but really at a physiological level, primarily to sense protons. And they're named GPR4, GPR65, and 68. And what's really interesting about this little family of receptors is that they're tuned to respond to slightly different pH 50. So for example, GPR4 responds to pH value around 8, whereas GPR68 responds to pH values kind of more acidic values around pH 6.5. So we became really interested in this. There's been a lot of work over the past many years trying to understand GPCRs that are activated by small molecule stimuli or protein hormones or peptide hormones. But this to us represented kind of a limit of the smallest stimulus that a receptor might be able to recognize. And we thought, okay, this is a pretty interesting problem. The challenge, though, at the outset is that most of the structural biology methods that we use in order to understand how a stimulus turns a receptor on don't really work for a stimulus like proton, because we can't see protons, barring some very advanced methods in X-ray crystallography that basically have never worked for GPCRs. We basically can't see protons. We can't actually see the stimulus here. Okay, so we wanted to get at this problem. Nick Hoppy, a graduate student in my lab, initially started off this work, and he came up with a very simple initial experiment. The idea was, what if these GPCRs worked like many of the other proton-sensitive proteins that have been found in the genome? Often these are things like ion channels. And in those ion channels is often a single titrated row, a single area of those proteins where a proton will bind and cause a conformational change. So Nick figured, okay, well, if that's true, then if I swap parts of this receptor GPR4 that responds at pH 8 with another parts of this receptor GPR68 that responds at pH 6.5, I might be able to switch the responsivity of one receptor for the other. So that's what he did. He basically took GPR4, swapped in various loops, extracellular loops of GPR68. And what was surprising to us is that it wasn't really until he started to change really large parts of this receptor, swapping one for the other, that you would get that he saw a relatively large change in the pH 50, like where this receptor would respond. And it's really not until you basically swap the top half of one receptor for the other that you basically start to get a pH response value that starts to approximate that of GPR68. So this is, again, a very crude, very simple experiment, but it started to teach us that perhaps the way that these proton sensors recognize protons is not by a single site, but it's really distributed across the entire top half of these receptors. So that was an interesting experiment, and again, we wanted to really understand how can we see very unconventional stimuli like protons. And at this point, another graduate student, Matthew Howard, joined my lab and jointly mentored by Willow Coyote Maestra, at the time faculty fellow, now faculty member at UCSF. And we decided we would probably need unconventional methods in order to figure out where protons actually activate receptors. That's what I'll tell you about. So as is common, at this point for the GPCR structural biology field, we were actually able to get structures of these proton sensing receptors. I wouldn't say quickly, but relatively readily. So Nick determined structures of GPR4, GPR65, and 68, all three of these proton sensing receptors, bound to these mini G proteins and these various G protein components that are pretty now common in the field. But the challenge again is that, okay, so we have these structures now. We can model these proteins pretty well. We can see, for example, that all of these receptors on their extracellular face, which is what you're looking at the bottom here, have a huge number of titratable residues, things that could potentially be protonated. But these structures basically don't teach us anything about where the protons act, right? So this is an example, again, of how structural biology often at this point is starting to give us information, but not really insight at the rate that we'd hope to get insight. So, okay, so we wanted to come up with a different method to get at this question. And in an ideal world, what we do in the structural biology field is we get a structure, and then we go make a mutation, right? We go make one mutation. We ask, okay, does it influence the ability of the receptor to signal? Does it influence the ability of the receptor to bind a ligand? But really, if you have 30 or so titratable sites, it's kind of a tall order to ask Nick or Matt to go say, okay, now make all 20 amino acid mutations at all 30 of those positions and try to understand how protonation works, right? That's really a tall order. But if you really want to understand from an unbiased perspective what sites on this protein are truly proton sensitive, that's really what you need. You need a way of covering mutations at every single position on the protein and be able to assess the effect of those mutations. So that's the kind of assay that we wanted to set up, and that's what Matt worked on. Now, the way that we wanted to do this is create a pooled assay. But first, we need an assay. We need a really reliable way to be able to detect GPCR activation, not by classic methods like, like a cyclic AMP assay or a glow sensor assay or these various Brett assays, but instead we need it to be a transcriptional assay. And for those of you who have played around with this kind of stuff in the past, you'll recognize that often it's very hard to get good signal to noise in GPCR transcriptional assays. So here's a simple circuit. Many of you have probably played with stuff like this. Here's a GPCR, it's GS coupled. It activates adenyl cyclase, causes production of cyclic AMP. Cyclic AMP via a cyclic AMP response element, in our case, produces a little GFP. Right? This is pretty simple. The challenge for the field for a long time has been that if you do this kind of thing, the basal activity of most GPCRs leads to very high production of whatever this reporter gene is, either a luciferase or a GFP. And so that's really what's been very challenging for some time. And so what Matt figured out are a number of tricks to really drive the signal to response profile of this assay much, much higher. A couple of things were needed. One, we were lucky to collaborate with Justin English's lab at the University of Utah. They've been developing new cyclic AMP response element and broadly transcriptional response elements for many GPCR pathways. And Justin had identified a cyclic AMP response element that had much higher signal to noise than what had existed previously. For example, what you get from Promega. So that's one trick. Another trick that Matt worked on quite a bit was to really come up with ways of dialing in expression levels of GPCRs at the cell surface, really trying to remove excess receptors that might drive basal signaling. And then finally, I think this is a trick that really ended up being quite important. Matt basically figured out that he could take GFP and couple it to a degron such that production of the GFP or production of the signal would require addition of this molecule TMP. In the absence of TMP, the GFP is degraded. And so what this allows us to do is we add TMP at the same time that we add the stimulus. And so we only integrate the transcriptional response during the time that the ligand is added. And this really ends up making a really big improvement because we basically only integrate over that small period of time. So with that, on the right here is the output of this assay. It's a simple flow cytometry experiment. We're looking at the production of GFP as a function of agonists for the beta receptor. And you can hopefully see here is that in the absence of an agonist, there's a very small population of cells that's on. We can turn these cells off by adding the inverse agonist ICI for beta receptor. Alpranolol, which has been thought to be a neutral antagonist, actually is a very weak partial agonist. And this really famous agonist, BI 167-107, really turns this population on to the same level that forskolin, that directly produces cyclic AMP does. So with this kind of approach, Matt also validated that this could work for GPR68. Here he's showing a dose response now, looking at the proportion of cells that are GFP positive as a function of pH. And what you'll hopefully recognize is that there's a very nice dose response showing that we can basically do pharmacology of this transcriptional assay. Okay, so why do we need a transcriptional assay in the first place? What do we want to do? Okay, the premise is we want to assess the effect of every single mutation that we could make on GPR68 in a pooled manner. And if you calculate out the numbers, it's about 10,000 different mutations. So how are we going to do this? So the way that Matt set this up is that he created a library of GPR68 variants, where at every single position on GPR68, we encode either a synonymous mutation for the wild type amino acid, or every single one of the 20 amino acids, as well as insertions and deletions. So we make a library like this, and that ends up being about 9,500 design mutations. And so now we want to test the effect of each one of these mutations with this transcriptional assay that I showed you. How do we actually do that? And the way this works is that we can then introduce constructs for each one of these mutants into an HEK cell line. And the way the cell line works is that it has a single site where the gene for the receptor can incorporate. So each cell now incorporates a unique receptor in its genome. And then we can pool all of these cells together and together turn these receptors on with the pH stimulus. And that will then give rise to a flow cytometry experiment where we'd get various levels of GFP. Again, some cells will respond better because they have a gain of function receptor. Some cells will respond worse because they have a loss of function receptor. So this allows us to ask for 10,000 different mutations, 9,500 mutations, what the effective mutation is on function. This is a general field called deep mutational scanning, and really Will Coyote-Maestras is really the pioneer for doing this for membrane proteins and GPCRs in particular. We've been lucky to collaborate with him to get this set up. Okay, so what does the data from this look like? This is one of these heat maps that you'll see from many of these deep mutational scanning papers. The way that you can read this is that you can go from the end to the C terminus. And in red are positions where if you mutate them, you get a loss of function. And in blue are positions where if you mutate them, you get a gain of function. But you can see hopefully that the C terminus has kind of a salt and pepper appearance, really not a lot of information there, likely because mutating there doesn't have a profound consequence. Whereas mutations in the transmembrane regions, as you'd predict, have pretty profound consequences for function. But at this point, this is kind of messy. What are you supposed to glean from this? If you look at really conserved motifs like the DRY motif, which is right here, or the NPXXY motif, which is over here, it's kind of buried in a sea of red. And for those of you who've been in the field for a little while, you'll recognize, okay, well, you did a signaling assay, but you didn't really control for surface expression. And so we realized is that if we really wanted to extract information from this assay, we would have to find a way to actually normalize for surface expression. So I'm going to tell you a little bit about how Matt did this. So Matt repeated another deep mutational scan. Now, in this case, not looking at the amount of cyclic AMP that had been produced, but instead how much receptor actually made it to the cell surface. And this is very simple. He basically just uses an antibody to the N-terminus, in this case, of a flag tag, and then does the same kind of sorting experiment. Okay, so we know as a gestalt in our field that the surface level of expression of any GPCR will influence the amount of cyclic AMP that might be produced, right? Just by virtue of how much receptor gets to the surface, things like receptor reserve, all of that. But how do we estimate that in our assay? And so the way that Matt came up with doing this is that he figured that the synonymous mutations that we had introduced in our library would have a distribution of surface expression levels just by chance. And that surface expression distribution could also be correlated with the amount of cyclic AMP that the synonymous mutations might make. And so each one of these black dots is one of these synonymous mutations. We could fit then a regression line to the synonymous mutations. That's basically a function that says, if we increase surface expression by X amount, what's the amount of cyclic AMP that might be produced? And now true gain of function mutations are ones that are as far away from this line towards the top. These are mutations that, given a level of surface expression, actually still produce more cyclic AMP. And conversely, the ones that are below that line are ones that are loss of function. So this is what the primary data now for our screen looks like. In black, again, are all the synonymous mutations, and gray in black is a regression line. And these mutations up here basically define positions that basically either have a gain of function and the ones down here have a loss of function. So coming back to this heat map that I showed you earlier, you can see there's a lot of different, again, a sea of red. But if we use this surface expression adjustment, we basically get a map that looks like this. And so hopefully what you'll recognize here is that some of the key positions like the DRY motif, or for example, in this case, the NPXXY motif, which is right here, really starts to shine out in red, right? So if I'll toggle back and forth again, you can see that now we can start to get actually position-specific information. Okay, so what do we do with this kind of information? Really, the goal is that this gives us an unbiased, although noisy, somewhat noisy, gives us a truly unbiased perspective on every single position on the receptor and how it might be important for this receptor's function. And so we can do now is start to map these mutations, the effect of these mutations back onto our structure of GPR68. Here now I'm showing you again our cryo structure of this receptor. And what we've done is colored in red mutations that basically cause a big loss of function. And so those are shown here. You can start to see that they start to coalesce around regions that are important for the kind of classic orthosteric ligand binding pocket of GPCRs. But you also see a lot of red in areas that interact with the G protein. These are things that we know now, from an era of structural biology to be really quite important. But from an unbiased perspective, it's really gratifying to see that the screen directly tells us these positions are quite important. Now, in the context of pH sensitivity, what we found especially nice is that 2 histidine residues immediately pop out of our screen as being really quite important for function. And this is what the primary data here looks like. You can see for this position, H269, substitution to positively charged amino acids like lysine or arginine is associated with a big gain of function. Again, telling us that introducing positive charge in that area is important for turning this receptor on. With XP Huang and Brian Roth's group, we were able to validate some of these. So here again is the dose response for the histidine residue. If we mutate it to a lysine, we get a left shift. Mutate it to an aspartate, we get a right shift. Okay, so that's one perspective. We can also paint this kind of information on to things like AlphaFold or other ways of predicting inactive state structures. I'm going to give you one little vignette of what we've learned there. Here again is our active state structure of this receptor painted on our loss of function mutations. And what we notice is in the AlphaFold prediction, this region makes a contact where our deep mutational scan indicates that mutations in the site cause a gain of function. So one could imagine that pertination of this glutamate residue, for example, drives conformational breaking of this region, and that's what then gives rise to transition to the active conformation. And just as conformation, if you mutate that glutamate to glutamine, trying to mimic that protonated state, we see that this receptor now becomes much more responsive to protons. So with that, we can start to kind of build a broader perspective and try to put together both the gain of function and the loss of function mutations and how they might influence receptor activity. And we think from that, you can kind of trace out a pathway of protonation events that basically cascade from the extracellular side of the receptor down to these common motifs known to be important for GPCR activation. And so, obviously, this has taught us quite a bit about how these proton sensor receptors work. But Willow's lab, we're quite excited to think about using these kinds of approaches, not just to understand fundamental mechanisms of how GPCRs function. We're really interested in now doing this, for example, for different pathways for GPCRs, for other receptors, for allosteric modulators, all of that. But at a different level, we're excited about thinking about how genetic variation in GPCRs leads to variation in drug response. Is a really nice graphic from Alex Hauser now a couple of years ago, trying to highlight the pharmacogenomics of GPCR drug action. And we think that these kinds of massively pooled parallel assays would allow us to start to get at how does the diversity of GPCR sequences in the population give rise to variable effects of molecules that are used in the clinic. Okay, so that's the first vignette about GPCRs responding to very unconventional stimulus. For the second half or so, I'm going to talk about another area of the lab that I'm really quite excited about. That again recognizes a very unconventional set of stimuli, and this has to do with with sensory biology. So it turns out that the 3 out of the 5 cardinal senses that we all learn about in grade school are encoded and enabled by G protein coupled receptors. Of course, for a long time, the poster child for understanding sensory biology and also GPCR biology has been the primary sense of vision. And what's cool about vision is that it's enabled by this very specialized G protein coupled receptor rhodopsin. And what I'll venture is that at this point, we understand an enormous amount about how rhodopsin works, not only at the level of the structural biology, but also the biophysics of rhodopsin, and also the neuroscience of the visual system. We have a deep understanding of how, for example, this one protein rhodopsin tunes the area around the chromophore retinal to be able to be tuned to specific wavelengths of light, and then how different wavelengths of light are recombined in the retina and in the brain to give rise to color perception. So we have this great way to connect the physics of light with the molecules that enable us to sense light and the neurobiology of light. And for other sensory systems, of course, we're starting to gain traction. Coming from UCSF, of course, I have to give a shout out to work from David Julius and Yifan Cheng here, describing how this molecule, for example, TRPV1, enables us to sense heat and molecules like capsaicin. But by contrast, the sense of smell has been far more challenging to understand. And part of the reason for that is that our sense of smell is tasked with recognizing a really enormously diverse set of molecules. And that's reflected by the fact that, again, about half of the GPCRs in our genome are just dedicated to the sense of smell. Here's just a smattering of a very small smattering of the kinds of molecules that we can detect by our sense of smell. But what fascinates me is not just the ability of our sense of smell to detect these molecules, but also to discriminate very small differences. So, for example, here are 2 enantiomers of an odorant called carvone, both R and S carvone, and one of these is experienced as spearmint, while the other one is experienced as a spice caraway. Now, of course, the big breakthrough in this field came about 30 years ago, pioneering work by Linda Buck and Richard Axel, where they discovered that a very large family of G protein coupled receptors are specialized to recognize odorants and are expressed in the nose. And it turns out there's 2 broad families of odorant receptors, so-called fish-like receptors. These are receptors that likely originated much older in evolution. They recognize water-soluble odorants. And in tetrapods that live on terrestrial climates, terrestrial environments, there's a huge explosion of so-called class 2 receptors. These are receptors that recognize odorants that can traverse through the air, often much more hydrophobic molecules. And with that, the logic of olfaction has really been set for about 25 or so years. This is what work, again, put forth by Linda Buck now quite some time ago. The model is that we can sense tens of thousands or maybe even millions of different odorants, but we don't have millions of different receptors. We have 400. So how does this work? The model is that a given odorant can activate multiple receptors, and that a given receptor can be activated by multiple odorants. And instead of a one-to-one relationship, it's really this combinatorial code of which sets of receptors are activated that the brain reads out to give rise to the perception of smell. So the logic of how this works is really quite different than a lot of the stimuli and receptor pairings that we're so used to understanding. There's not, for example, these are sets of hydrogen bonds, these sets of keys that interact very precisely. It's much looser, and the logic for this has been really quite hard to crack. So this is a problem that we've been after for some time, and what's obvious is that, like, at some level, this is really a chemistry and a structural biology problem. And we've made such enormous advances as a field trying to understand the structural biology of G protein coupled receptors at this point. It turns out that odorant receptors in particular have been really, really hard to work with. And a key reason for that is that even among GPCRs, they're incredibly poorly expressed. On top of that, their ligands have incredibly poor solubility. And like many other GPCRs, are probably even worse, so these receptors are incredibly unstable. So we ended up, we decided to take on a new collaboration and try to tackle these receptors. And this has worked with Hiro Matsunami at Duke and Vaidehi Nagarajan at the City of Hope. Vaidehi, many of you will know, has worked on GPCR field for some time. Hiro is really a leader in the olfactory receptor space. In my lab, Christian Billisbola, a postdoctoral fellow at the time and now a senior scientist, and Claudia Aldinas Del Torrent Massax have been driving this work. And in Hiro's lab, Claire, Demarche, and Jeevan have been driving the work, and Vaidehi's lab, Wynand, Belden, and Ningma have been driving this work. Really a highly collaborative effort that I'll talk about today. So we figured, okay, we can't make most odorant receptors because they're basically impossible to make, probably because there's some chaperone in the nose that allows these things to fold. But there's this percolating literature for about the past decade or 15 or so years that many of these receptors can be expressed outside the nose and have moonlighting functions. They can do other things in other parts of the body. Here's a nice report from about 11 or so years ago looking at transcript levels of various odorant receptors in other tissues. And this group and many others have recognized that these receptors perhaps are truly expressed in other areas of the body. And there's, again, on the right, I've just put a little figure that tries to highlight various biology that people have implicated these ectopic receptors in. So we figured, okay, these ectopic receptors may not recognize the chaperones that are in the nose and might perhaps be more tractable for structural biology purposes. And on top of that, we decided to focus on receptors that recognize very soluble odorants, things like propionate or lactate shown here, as compared to these very hydrophobic odorants. And then finally, to deal with the stability problem, one trick that my lab has been using quite a bit are these mini G proteins. Really originated by a seminal paper from my late UCSF colleague Henry Bourne, this paper from about 30 years ago, where they showed that the alpha subunit of a G protein can be split in half by so-called a Ralph domain and a Gale domain. And of course, in the modern era, Chris Tate's lab has engineered these so-called mini G proteins that remove this flexible alpha helical domain and engineer this receptor, this protein to be primed to interact with GPCRs. And our simple observation after many years has been that these inactive or these receptors that are on their own are quite unstable. But for most GPCRs, if you couple them to a heterotrimeric G protein, they become really quite stable. So we figured, okay, if we fuse one of these receptors to one of these mini G proteins, maybe that will make it more stable. And we found that trick to be quite useful in the lab for a variety of receptors. So with that, we focused on one receptor, again, one of these fish-like receptors, using a variety of kind of common methods now in the GPCR field to get complexes of these. Again, the exciting thing about modern cryo-EM is that we can get away with very tiny quantities of protein. Again, the protein that we worked on, this protein OR51E2, really still quite challenging to work with. But Christian in the lab was able to determine a structure, in this case, the first structure of any vertebrate odorant receptor to pretty reasonable resolution. So this is what it looks like. Again, it looks like many other GPCRs. But what's exciting is that it's, again, bound to an odorant molecule. So where does this odorant bind? Basically overlaps pretty similarly with kind of the classic orthosteric pocket of many GPCRs. This is a comparison where adrenaline binds the beta receptor and where propionate binds its own receptor. And I'm going to delve into a little bit of minutia, but I think it's going to be important for trying to understand evolution of the olfactory receptor. So please bear with me for some of the minutia here. Now, one thing that we wanted to understand is this receptor recognizes a very, again, soluble odorant, in this case, propionate that has this carboxylic acid. It turns out these so-called fish-like receptors all have this conserved arginine residue that in our structure basically comes back and binds this carboxylic acid. And in simulations done by Vaidehi's group, we could show that this contact is maintained over many microseconds of simulation. So that gives us one idea about how this specific set of odorants is recognized by this set of odorant receptors. Now, a big question in the field of olfaction is what drives the tuning of a receptor to recognize a specific set of odorants. And we want to start to get at this question. And the way that we did this is we looked at the kinds of odorants that this receptor OR51E2 responds to. Turns out that it only responds to very short chain fatty acids, things like propionate or acetate. And if you start adding more carbons, basically it's not responsive. And the reason for that is that it has a very small binding pocket. So a very simple experiment we could do is, okay, just simply make more space in the binding pocket on the back end. And again, it's a cute experiment, but it starts to build intuition for how the shape of the binding pockets of these odorant receptors give rise to tuning for various kinds of odorants. For example, if you introduce an alanine at this position where phenylalanine is, now this receptor starts to respond to longer chain fatty acids. Again, simple kind of structural biology experiment, but starts to build intuition for us. Okay, so that's one receptor. What I told you is there's hundreds of receptors, and we can't make any of them. Like, that's the only receptor Heroes Lab has literally tried all 400. It's the only one that expresses at any reasonable level. So we decided to take a slightly different approach to complement our initial efforts to understand the structure of odorant receptors. And that is a method that many protein engineers use, which is ancestral protein reconstruction. So the idea here is that if many of the modern sequences of whatever you're trying to work with aren't really tractable for some sort of protein engineering project, often what protein engineers will do is they will infer an ancestral protein sequence or come up with a consensus sequence. And often these sequences are more thermostable, promiscuous, or evolvable. So we figured, okay, this was an idea that Heroes Lab had been working on for some time. And I'll just kind of give you a premise of how they do this. So the way this works is that they line up, all of the submembers of one of these odorant receptor families. So there's about 16 or 17 of these large families of odorant receptors in humans. We line up, the 51 family, which is comprised of about 23 receptors, and then we just make a very simple construct, which is a receptor that keeps the most common amino acid at each one of these positions. So, and this is so-called consensus receptor. Now, if you look at a dendrogram, this consensus receptor lies at the root of the tree. Again, consistent with the fact that it's just the most common amino acid at each position. So that's all fine. This has been done in the past for other soluble proteins. But what's really remarkable is that these receptors express at remarkably high levels. So what you're seeing here is just a simple experiment looking at flow cytometry of HEK cells expressing each one of these 23 family members, as well as this consensus receptor. In the black here is just the vector-only control. You can see that, each one of these, in gray lines, each one of these individual one of these family members, you can see most of them just don't express at any level. This is really the challenge for the field. We cannot actually detect surface expression of the vast majority of odorant receptors. You can see why we worked on OR51E2. It's one of the only receptors that expresses at any level that's measurable. And by contrast, this consensus receptor expresses at levels that are very compatible with structural biology that many people are comfortable doing at this point. So we then said, okay, this is admittedly a Frankenstein receptor, but what can we learn from it? So we determined a structure of this receptor, this consensus OR51 receptor, and we knew from signaling studies this receptor is constitutively active, so we captured it without an odorant bound in the binding pocket. Okay, so is this Frankenstein receptor any good? So we start to compare this receptor, this consensus OR51 receptor, with one of the family members, in this case, OR51E2, the truly human receptor that we determined the structure of. At the secondary structural level, they're basically identical. You can't tell them apart. Maybe slight differences in TM6, but otherwise, pretty darn similar. But if we even just zoom into the binding pocket, the area where the odorant binds, you'll see hopefully that in blue is this consensus receptor and green is a native receptor. Then most of the residues are obviously conserved, but also the positions of those amino acids are highly conserved. So for example, this arginine, even though there's no odorant bound here, is poised and ready to interact with that carboxylic acid. So, okay, so what is this good for? We think that these consensus receptors give us a really great starting point to do homology modeling of the broader family. So for this specific case, this consensus receptor, we think could allow us to now homology model a cousin of this green receptor called OR51E1. And this receptor, unlike 51E2, actually responds to longer chain fatty acids. And we could use this homology model to rationalize, for example, why OR51E1 prefers longer chain fatty acids like pentanoic acid. In this case, it's the docked structure of that. So with that, I told you a little bit of validation of understanding these fish-like receptors. Really what we're after is trying to understand the diversity of these tetrapod-specific receptors. These are the receptors that recognize cool stuff like grapefruit or mint or caraway, all these amazing odors. And so we've been using the same consensus strategy to go after these. And here, for example, is another consensus receptor we designed for the OR1 family. What's interesting about this consensus receptor is that it recognizes a huge diversity of odorants, really broad classes of molecules. And we were then able to get a structure of this consensus receptor in the presence of an odorant L-menthol. Again, now representing kind of a more classic odorant. So we've done this exercise for a variety of consensus receptors. And the punchline is that we think there's kind of a conserved mechanism across these class 1 and class 2 odorant receptors. Although the specific motifs are different, the way that odorants bind and turn these receptors on is perhaps conserved. So let me tell you a little bit about what we've learned there, and then I'll zoom out a little bit and give you some speculation on what we think this might represent for the evolution of olfactory sense in vertebrates. Okay, so returning back to OR51E2, the receptor for which we have the most information, the native receptor, we noticed that, again, this arginine residue interacts with this carboxylic acid, and that's really critically important for turning this receptor on. We could do a simple simulation experiment, just remove that ligand, that odorant, and what we notice is that this arginine residue starts to flop in the breeze, and that's associated with dynamics of this entire loop, this extracellular loop 3. Now, we could design a very specific experiment. If we were on the right track, we could design a gain-of-function experiment, introduce a mutation in the binding pocket that would pin back this arginine. And if we're on the right track, that should be a gain-of-function to be constitutively active. And so that's what we did. Here's the simulation data showing that if we introduce a carboxylic acid at a very specific position by mutating a glutamine to an aspartate, we can predict that this arginine would come back into the binding pocket, stay persistently there. And what we show here is that even in the absence of propionate, this mutant is basically constitutively active. So our model for how odorants activate odorant receptors, is a relatively simple model. We think that this helix TM6 is relatively dynamic in the absence of odorants, and that's associated with dynamics in the extracellular loop 3. And that what odorants do is they make interactions with TM6 and many other interactions that does two things. One, by pulling on the top of TM6, there's a pivot point kind of in the middle of these receptors and causes a conformational transition on the inside that allows G proteins to interact. But also, it causes an ordering of this extracellular loop 3. Now, this was speculation from our work because we didn't have an inactive structure of OR51E2. And really beautiful work from Hee-Jung Choi and colleagues determined a true apo structure of a consensus odorant receptor, where they showed that this mechanism is exactly spot on. And you can see from this image from their paper that that these transitions are exactly end up being exactly what we proposed earlier. Okay, so what does this mean? This is a speculative part. Would love to get feedback, but also bear with me for a minute here. We want to understand how a single family of receptors that's exploded in evolution can recognize such a huge diversity of odorants, but still likely have the same activation mechanism. They still have a lot of the similar motifs. They're not distant like a beta receptor and rhodopsin or something, right? Like they still have very similar activation motifs. But how does a single family of receptors deal with such a huge diversity of chemistry? So what we did is started to look. Again, we're not going to get structures of all of these. So we started to obviously turn to AlphaFold, like many structural biologists and many biologists in general do these days. And so what you're looking at here is a compendium of all 400 or so human olfactory receptors and their AlphaFold predictions. You're looking at it from kind of like the outside looking in. And it's colored by the AlphaFold confidence metric. So in red are regions where AlphaFold feels very confident, and blue are regions where AlphaFold doesn't feel very confident. And what I'll say is that regions that are red here are really quite well predicted at a secondary structural level, but they're really quite divergent in sequence. It's not like these regions are almost always the exact same sequence. And in particular, what fascinated us is that this specific loop, this extracellular loop 3, AlphaFold has poor confidence in and is likely to be very structurally heterogeneous. So what do we think this means? The speculation here is that we think that across evolution, diversification in the structure of this extracellular loop 3 allows odorant receptors to mix and match and interact with a lot of different types of odorants, but that then on a single receptor, binding of an odorant causes structural rigidification of this part of the receptor, and that leads to a common activation mechanism. So it's this way of pairing the need to have huge diversity of chemical interactions with a common activation mechanism. Now, I would venture that olfaction is probably one of the most challenging chemical recognition problems for biology. This one family of receptors has to grapple with a really huge diversity of chemistry in natural environments. And perhaps if we look for inspiration, where else biology has to solve this problem, I'd venture the adaptive immune system is another great area, right? So your immune system is tasked with being able to recognize antigens that could be infinitely dissimilar to each other. And so if we kind of think about ways in which biology tries to solve these problems, what I propose to you is kind of a central mechanism where olfactory receptors do this. We think there's a lot of parallels to how the adaptive immune system does this, right? So for example, here on the left are structures of, in this case, single domain antibodies, but antibodies as a class do this as well, where diversity in the shapes of a single loop, in this case, the CDR3 of antibodies, allows that protein fold to recognize such a huge diversity of antigens. And we think and a kind of a similar logic applies to how olfactory receptors diversify a certain region of the receptors in shape, and that's what allows them to recognize such a huge diversity of small molecules. Now, in the one case, this is evolution for odor receptors that happens across species over millennia, and on the left is obviously evolution that happens in a single individual through somatic hypermutation. But we think the principles are somewhat similar. So with that, our work on olfactory receptors continues. We have a lot more to do. Again, our long-term goal is to use a combination of structural biology, but also a lot of the methods I described in the earlier vignette, massively pooled assays, to really take this cartoon diagram and actually have a predictive model for how a given small molecule odorant interacts with some subset of receptors. And we certainly won't be the only ones to make headway on this. It's a lot of other people that are working in this space. But hopefully the dream over the next some number of years would be to really have a way of predicting what the smell precept of a small molecule odorant might be. So with that, I'll wrap up and just highlight that a number of phenomenal people in my lab have led the work. I think I give a shout out to all the people that led that. We have a really, really collaborative team, a lot of people that work deeply with each other to try to drive this work forward. And we're really lucky to have support from the NIDCD for all of our olfactory receptor work, and from a variety of foundations for some of our more exploratory work on proton sensing receptors and these new massively parallel assays. That happy to take questions on this work or other work. Yeah.
Thank you very much, Aashish, for this phenomenal talk. We already have a hand raised. If you want to unmute yourself, let's start with, yeah.
Hey, Aashish. Eli McDonald. I'm a postdoc in the Babu lab, and you mentioned kind of at the beginning that there may be a chaperone or set of chaperones expressed in nasal cells that are responsible for expressing these olfactory receptors at high levels. Do you have any idea what these chaperones might be, or is there a way to discover them? I mean, if we could co-express these, would this be a way to increase expression so that you could work with these?
Great question. So, Hiro Matsunami's lab discovered some years ago some of these chaperones are called receptor transport proteins, RTP1S and RTP. They have some effect in heterologous cells like HEK cells, but they're still not really very robust chaperones. We think that there's likely other chaperones just remain undiscovered, and so we're doing CRISPR screens in collaboration with Rebecca Voorhees at Caltech to try to find perhaps new chaperones that might help in that kind of context, and a little bit of work with Stavros Lombardis trying to actually think about how to look at this in primary olfactory sensory neurons. But there's still a lot of work to do to figure out what is the biogenesis machinery in olfactory sensory neurons, and then is there some way to actually port that over or find new machinery in heterologous cells that might allow these receptors to get exported in a much more efficient manner.
Thank you. I hope that answered the question. There's another question, Libin, if you want to unmute yourself and ask.
Sure. Hi, Aashish, this is Libin. So I have a question actually for the first part. So it's very interesting. So I look at your PhD sensitive receptor that you swap different domain of the receptor. So when you do this swap, you are going to swap the transmembrane domain as well, or just the extracellular domain and intracellular domain?
Yeah, the cartoon maybe isn't appropriate. So what we do is we swap maybe the top 1/3 of like the transmembrane domain along with the extracellular loops. You just find this image again. The cartoon tries to make it a little bit simpler than the experiment actually is, and it's a little bit hard to show the actual, exactly where the cut sites are. But it retains some of the regions that we thought going in might be important for proton sensing, which are not just.
So you see that there's in the middle transmembrane domain there are some common allosteric pathways they are conserved, right? The mostly this kind of the pH sensitive. It's dependent on the how to say the maybe depend on the protylation or depolylation amino acid in this region, such as you in your heat map you point out the histidine. Did you look at the cysteine or arginine or some lysine distribution in these domains in your heat map?
Yeah, good question. So our heat map is unbiased now. So this is the challenge we faced, right? Which is okay, you could say that you could have an a priori, a prior going into this experiment and say, okay, well, histidines are important, right? So it's going to be all histidines. But if you look at the actual titratable sites, there's a lot of them, and I didn't even introduce the fact that cysteines and tyrosines, other residues can be titratable as well. So, so if you and on top of that, it's very, very hard to predict local pKa's just from structure alone, right? It's basically almost impossible to do that. So that's why we ended up feeling it was necessary to do this kind of unbiased screen. I have to go back and look at and see if we found, for example, lysine or cysteine residues that we think are responding in a titratable manner. I think most of the mutations that we found in residues like that were kind of conserved GPCR motifs. Like we'd find, like a mutation in DRY motif or something like that, or the CWXP motif, the cysteine probably has an effect, but we didn't ascribe those likely to a protonation event.
Sure. Thank you very much. Yeah. Great work.
Sumit, if you want to go next with your question.
Oh, yes. Hi, Aashish. Thank you for the really cool talk. It's exciting to see these really unconventional problems being tackled. I was wondering if I could ask you about your first story with the odorant receptor, where you were doing some mutagenesis on it. And if I saw correctly, when you did there was a mutation of a phenylalanine, and when you mutated that phenylalanine, you gained sensitivity to long chain fatty acids, but it seems like you also lost short chain
very astute. Yeah.
Potency there. So I was wondering if you could comment. You mentioned there's some idea that these pockets could be flexible, they could accommodate a variety of ligands, but it seems like they're not hugely. I mean, you mutate one residue and you've completely changed your. I mean, from what I assume, if I am reading that correctly, it seems like you totally change your selectivity here.
The sensitivity for propionate does go down quite a bit, right? Certainly about a half log or so. What I ascribe this to is that propionate is really quite small, and you really need all of the ideal van der Waals packing on the back end to get any potency. I mean, the fact that it's actually has any effect is actually kind of mind-boggling because it's so small. So that's at least how I had rationalized it to myself, is that the small amount of van der Waals packing that you see here that's lost, likely with this alanine mutant, probably is responsible for that loss in potency. But in the absence of really deeply diving into what the dynamics of propionate in this mutant looks like, we probably wouldn't have a great idea. But that'd be my rough guess from the outset. Yeah. But maybe just to broaden out a little bit, I think there is this very clear phenomenon in odorant receptor binding pockets where single mutations can have really dramatic consequences for retuning receptors. This is kind of an edge case because these receptors respond to kind of a very narrow subset of odorants, which are carboxylic acids. But if you look at receptors that respond to receptors like this that respond to a whole diversity of odorants, in receptors like this, if you make one mutation, you actually completely retune the sets of odorants that they interact with, and the weights, their sensitivity to each one of these will change pretty dramatically. That's true in our work, and also in Vanessa Ruta who's been studying insect olfactory receptors, and she's shown that beautifully in that system too. So it's really hard to come up with kind of like this clear logic that we often have with other GPCRs. We make a mutation, we really understand like the effect that it has on a ligand. Because these are responding to many ligands at once, mutations don't have kind of a one-to-one effect. And because most of the contacts are van der Waals contacts, it's kind of a mess for thinking about what's actually driving interactions and then encoding information downstream.
Thank you very much. Michael, do you want to ask your question too?
Sure, yes. Hi, Aashish. Great talk. I was very excited to hear about it. And I'm interested in the consensus receptors. So it was very striking and fortunate that the consensus receptors had a much higher expression. Were you expecting that? Or is that something that's expected for these consensus type of receptors? Or is this just a fortunate thing that happened to work out for this particular receptor?
So the consensus idea is quite old. And we didn't come up with that, right? So that I think the earliest papers are decades old now for enzymes and for immunoglobulin proteins, that if you make a consensus that often is much more stable and has these properties that individual extant members of sequences don't have. And it's been applied to a couple of membrane proteins. I think Hiro Matsunami's kind of just really Hiro's idea was that perhaps odorant receptors might be particularly amenable to the strategy because the premise of his thinking is that odorant receptors are evolving very rapidly without strong evolutionary constraints for folding, and that perhaps a more ancestral odorant receptor, was a little bit more functional and folded, and if we could try to recapitulate that kind of ancestral state, that might have these properties. So in general, consensus proteins do show these benefits, but I think the huge gain here may reflect something unique about kind of how lousy odorant receptors are for folding in the first place.
Okay, interesting. Thank you. And if you're looking for chaperones, then could you look for proteins that were evolutionarily coupled with that? If you have this one consensus sequence that's stable, doesn't need a chaperone, it seems. So that chaperone would have to evolve after the shift, right? I'm just
I don't know how to do that, but it's a good
idea. I don't know
idea.
either.
I don't practically know how to do that. Yeah.
Just spurred my question. All right, thank you very much.
A bit of speculation is also always nice. We have one more question in the chat from Said. Is there an effect of membrane lipid composition on olfactory receptor selectivity?
Ooh, it's a good question. I don't know how to answer that question, unfortunately, but it's a good question. Almost certainly there is, but there's not really a great way to do that experiment. I'm assuming this is selectivity for the ligands. There's not a lot of G protein selectivity for odorant receptors. They all, at least in sensory neurons, coupled to G olf. So that's probably not that variable, but it is possible that different lipid environments could change what odorants a receptor responds to. I don't have a great model system, and we haven't particularly looked at that.
Thank you. Oh, there's another one. Aditya, go on.
Hi, Aashish. Really nice talk. I was wondering, how do you think about consensus sites for lipids versus consensus sites for protein interactions? Do you weight them differently? Because it seems like, especially in case of lipid consensus sites, lipid interaction consensus sites, they appear very randomly everywhere and might not be really relevant.
Yeah, good question. Okay, so I'm embarrassed to admit that we have not thought at all about where these mutations are and where we're putting these. Literally, it's as simple as we just line up the sequences and just make a consensus construct. There's no more thinking beyond that, right? I think the next level of this is to do what you're starting to indicate, right? Are there areas of this of where the consensus is having its effect, right? Is it a lipid binding site? Is there a certain contact that's on the inside of the receptor that's better maintaining these consensus constructs? We haven't gotten very far with that. We've tried some experiments, but haven't really pinned down this is an area that the consensus proteins are really optimizing, and that's why they're so much better. if we had that kind of information, I think we could start to get at what you're talking about, which is there a lipid packing defect that's, you know, improved and that's what's important, or is it something, you know, inside the receptor that's more important? Yeah, it's a great question. We just don't have a real answer to that.
Thank you.
Thanks to all of our the people who asked questions, and thanks to Aashish. I think before we close, I did have kind of one final question, because this is a symposium for early career investigators, and we have